Okay.
Okay. Hello, everybody. Let's wait a few more seconds for everybody to join us. We're recording today's talk. Just a couple more seconds when people join us. Rosa, you let me know if you think we should get started.
You could start.
Okay, so as you know, everybody, most of you are regular attendees. This is the European Microglia Webinar Series. This is organized by 3 teams at the University of Lausanne, the team of Rosa Paulicelli, the Collège de France with Morgan Thion, and us ourselves at Atucaro, several PIs. We have a fantastic list of upcoming talks. Please stay posted. We just have to make an announcement because up until now we've been using this link that has been repeatedly being used for the past couple of years here in Zoom, but we are gonna change the format. So please be aware that in the future talk, we will be changing the link. We will be explaining this in the email, but of course, please be aware. So today's talk, we have a fantastic speaker. Rosa, if you can go next. Yeah, we have a fantastic team, research by Tara Silva, who will be talking about microglia as sculptures of white matter during development. And for today, it will be the team of Morgan Thion at the Collège de France, who will be introducing the speaker. That will be Marta Pereira, and the questions and answers will be handled by Sarah Vinier. So without further ado, please everybody welcome Tara. I'm just gonna say, please, most of you turn off your cameras because probably it will increase the signal. And then when we finish, you can either raise your hand or just type on the chat whichever question you may have. So please go ahead. Welcome, Tara.
Welcome, it's great to be here. Thank you.
Hello, everyone. So I'm gonna briefly introduce Tara. She's an associate professor at the Cleveland Clinic and Case Western Reserve University School of Medicine. She received her PhD from the University of Pennsylvania and completed postdoctoral training at Children's Hospital Boston, Harvard Medical School. Her research centers on how inflammatory responses contribute to neurodegenerative diseases such as multiple sclerosis and Alzheimer's disease, with a particular focus on how these processes impair neuronal function. A major direction of her laboratory is the development of therapeutic strategies that target the glutamate-mediated excitotoxic cascade, a key driver of neurodegeneration. In parallel, the laboratory investigates innate immune responses in the central nervous system, especially the role of microglia in clearance during development. So the floor is yours. Well, the screen is yours.
Thank you. I'm going to go ahead and share screen. Okay, great. Can everyone see my screen?
Yes.
Perfect. Okay, thank you so much. Well, it's a great privilege to be here today. Thank you so much for inviting me. Many people on this line, I have read your papers and admired your work, and a lot of your work has actually influenced us looking at microglia and their relationship to white matter refinement. So thank you for all of the great.
Tara, you muted yourself, so
Yes.
I think you muted yourself. So
Okay, can you hear me now?
Yes.
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 Sorry about that. So I'm going to try to advance my slide here. Okay, here we go. So as we all know, during development, microglia invade the developing brain during early postnatal development, making them perfectly poised to regulate neurogenesis and synaptogenesis. So we became very interested in whether microglia may also be regulating in oligodendrogenesis due to its time period when many mechanisms of neuronal refinement are occurring, especially since oligodendrocyte myelination plays such an important role in normal neuronal function. And the peak period for oligodendrogenesis is postnatal day 7 in the corpus callosum. So we were very interested in microglia OPC interactions during this early postnatal time point. So just so we're clear where we're looking, we are doing these studies with transgenic mice, with reporter mice, and we're looking at coronal sections of the brain where we can really see the full sort of envelopment of the corpus callosum. And this corpus callosum houses myelinated axons that form interhemispheric connections, mainly from pyramidal neurons in layer 5 and pyramidal neurons in layer 2-3. Also, the corpus callosum overlies the lateral ventricles, where actually we have proliferation of glial cells coming into the white matter postnatally. So unlike neurons that really sort of develop in place embryonically, oligodendrocyte myelination occurs postnatally. And we were very interested in the role of microglia interactions to sort of understand this refinement process and if it's occurring. And we used a reporter, a common reporter, CX3CR1-GFP, which is a fluorescent green reporter that's genetically knocked into the CX3CR1 promoter, which is a promoter for both microglia and peripheral macrophages. And what we found at embryonic day 14, similar to the work of Ganau and others, that really microglia are coming from the yolk sac and then entering into the brain during early embryonic development. And they're really sort of evenly distributed throughout the layers of the cortex and the white matter. However, at around right before term birth, so in mice, term birth is what we call P0, so it's about 21 days before a mouse is born. We saw a very, what I'll call just fat or amoeboid looking microglia accumulating in the subventricular regions of the lateral ventricle and eventually moving into the corpus callosum area. So Ashley Nemes, who was a postdoctoral fellow in the lab and worked on this project, and she is now an assistant professor at Case Western Reserve in the Neurosciences Department, showed that these amoeboid microglia really stay restricted to the corpus callosum or the white matter. We see very few amoeboid-looking microglia in the overlying cortex, and this sort of high power view shows that, whereas the cortical microglia have a more ramified look with branching, and the amoeboid microglia, which are found in the white matter of the corpus callosum, look very fat and what some people might mistaken as activated, but again, this is normal during normal development, so there's no disease state here. And what we did is we did 3D reconstructions using Amaris to quantify this engulfment. And this is just taking one of the Z-planes, which is about 0.3 micrometer. So just proving that what we're seeing, what are we seeing inside these microglia? So in this particular experiment, we stained microglia with IBA1 in green. And then we used a reporter for OPCs, or oligodendrocyte progenitor cells, using the NG2 CRE-ER TD tomato reporter, which we tamoxifen induce, and it turns all OPCs red. And then bisbenzamide is a marker for all cells. And what we clearly see is that OPCs are encapsulated within microglia, giving the appearance that microglia are actually eating whole oligodendrocyte progenitors during early postnatal development. And if we take a look of just a snapshot across this time period, again, these are 40x 3D reconstructions. This is postnatal day 4, postnatal day 7, which we have peak engulfment. And then by P11, we start to see, or P9, we start to see that there's less engulfment. And by P11, the microglia are starting to assume a more ramified morphology as oligodendrocytes are starting to myelinate. So really myelination begins probably somewhere around P11. And using the Amaris software and the CoLoc tool, and it's a method that we have published in CellStar protocols, we see that the peak engulfment is around postnatal day 7. and the contacting for microglia just contacting OPCs, not engulfing them, continues during development as that myelination happens. And so how did we come up with these two types of interaction, which I'll refer to? And one is microglia contacting an OPC and microglia eating an OPC. And this came from live cell imaging. So what we did is we made ex vivo slices of the brain and kept them alive for a 12-hour period, and then did 2-photon in vivo imaging across this time period to watch how microglia engulf these OPCs. Because if they're actually inside of the microglia, we wanted to watch microglia interact with OPCs, either contacting them or eating them in real time. So if we take a look at this video on the left-hand side here, what we can see is this microglia comes in contact with this OPC. It senses it, but then it says, you know, you don't taste very good. I'm going to move over to somebody else. And then over here, we can see that this OPC is being engulfed. So if we focus in on a microglia that's eating an OPC, we can see the microglia senses the OPC, and then when it decides to eat it, it does so in about 20 minutes. So the next question we had is, well, are microglia just eating these OPCs because they died? Because there's like, you proliferate too many of them, the ones that don't myelinate die, and now the microglia are eating the dead and just clearing the debris. And surprisingly, what we found is that the majority of engulfed OPCs, whether we're looking at perfusion fixed tissue or we're looking at live imaging, is that the majority were not dead before being eaten. So what we did is we used phosphatidylserine, which is a marker that gets expressed on the cell surface of stressed cells. We also used caspase-3, which is an apoptotic marker. We used PSVU that detects phosphatidylserine and other charged molecules that get expressed when cells are stressed, as well as we did live cell imaging with simultaneous staining with oxazole blue, which is a cell death marker, and DIL-C, which is a mitochondrial membrane viability marker. So we wanted to see if they're really not expressing these cell death markers, then they should be staining for DIL-C, which only gets intercalated into the microglia if the mitochondria are healthy and alive and viable. And what we found is two types of processes that are occurring. One is that 85% of engulfed OPCs are viable, meaning they're not expressing oxazole blue, they don't express the phosphatidylserine or PSVU, but they are positive for this DIL-C5, this membrane mitochondrial membrane potential. And about 15% of engulfed OPCs are dead before being eaten. So there's two processes that we're continuing to understand. The next question we wanted to ask is like, well, what role does this process really have? So if you block it, is there a physiological impact or not? Or if microglia are not able to eat the OPCs, that you just get normal myelination. So what's actually going on? So what we did is we started off using the fractalkine receptor knockout mice. These mice have been traditionally used to study synaptic engulfment by many of the people on this call. And so we thought we would look to see whether the same receptor is also being utilized by amoeboid microglia during early postnatal development in the white matter. And what we also know is predominantly the fractalkine receptor is expressed on microglia, and also the ligand for the ligand for the receptor fractalkine. So fractalkine receptor is CX3CR1. The fractalkine ligand is CX3CL1. It is expressed on OPCs and downregulated in mature OL, so suggesting that there is a developmental difference between OPCs and mature OLs, and could that somehow be involved in this engulfment process? And what we did find is, first off, when we look at the fractalkine receptor deficient mice compared to what we call wild type here, but actually these are the het expressing. So if they have GFP, this is the het expression of CX3CR1, and GFP is also expressed in the homozygous deletion of CX3CR1. What we first wanted to make sure is that because fractalkine is involved in chemokine signaling, that was there a change in the number of cells, the number of microglia green cells in the knockout versus the wild type. And we did not see a difference in the number. So the numbers were the same, because obviously if we decrease the number of microglia, that would influence engulfment in and of itself. And then number two, we wanted to make sure that we didn't change the volume of the microglia or the morphology of the microglia that would somehow indicate that something else is going on in these microglia. And what we found is that in the knockout mice, that even though microglia still came in contact with the OPC in the knockout compared to the wild type, that there was about a 50% reduction in the engulfment of these cells. So we then said, well, if the engulfment of the microglia engulfment of OPCs is reduced by about 50%, and if OPCs are alive before being eaten, then removing the ability of microglia to eat those OPCs should increase the number of OPCs. So we're not eating the OPCs, so that means, and they're not dying, so we should have an increase in number. And this is a concept that's been developed in the literature, that primary phagocytosis or phagoptosis is eliminating viable cells, has an important distinguishing feature in that if you block the engulfment, you prevent loss of the target cell. And this has been shown in some cases in development of neurons, and it's also been shown in immune cell development. So indeed, what we did find is that if we block the fractalkine-dependent microglia pruning of OPCs, it results in an increase in the number of OLIG2 and CC1 positive cells. And we've also confirmed that this increase only occurs after engulfment. So before engulfment, we have the same number of OPCs, but after engulfment in the fractalkine knockout mice, we do have an increase in OLIG2 CC1 here. And so the next thing we hypothesized is that can this engulfment, if we have an increased number of OLs, how is that going to affect myelin formation? And in fact, we were quite surprised because we predicted that since we had an increase in the number of OLs, that therefore we were going to get an increase in myelin thickness because we'd have more myelin. But in fact, we saw the opposite. So what we found is that in the wild type mice, they have thicker myelin, and in the knockout mice, overall, they have thinner myelin. And we look at the G ratio here. And if we look at the G ratio, which is the thickness of the myelin plus the axon diameter over the axon diameter, so we can look at myelin thickness per axon diameter, it seems it's pretty much evenly distributed. So there isn't one specific axon diameter that's being dysregulated or has dysregulated myelin. It seems like overall the initiation of myelin is happening, but it's just not efficient. It's not producing the same thickness of myelin. And we plotted this in different ways using relative cumulative frequency. We also showed that the axon numbers are not different. We have the same number of axons between the two, and the percent unmyelinated axons is not different from percent myelinated axons. So the same number of axons are being myelin, it's just that myelin thickness isn't the same, so myelination is inefficient. And there can be many reasons for that. We're thinking that we're starting to look at this with single cell, but that perhaps there's limited resources. So things like cholesterol and other myelin proteins that are necessary for proper myelination may be limited. And so if you have an overcrowding effect, you have limited resources. The other aspect we're looking into is if we have more myelin branching is shown to be associated with internode distances, and perhaps with shorter internodes, we have thinner myelin. So these are some opportunities that we're going to further explore as to why this happens. And then John Shelestak, a current postdoc in the lab, asked, well, if the amoeboid microglia are coming out of the lateral ventricles into the corpus callosum, what's happening in other white matter tracts during postnatal development? And this was a paper recently published in 2025 showing that here the postnatal infiltration of these amoeboid microglia happens concurrently in the corpus callosum and what's called the fourth ventricle. So as we know, our ventricular system is tied together with our lateral ventricles and our fourth ventricle, also our third ventricle, which I'll talk about later in the talk, that is leading to the optic chiasm. And what we find here again at postnatal day 3, this is around the fourth ventricle, we see these amoeboid microglia accumulating and then entering into the cerebellar white matter here, which is called the trunk of arborvitae. And we can see that they become highly proliferative and accumulating at postnatal day 7, which is the same time point that we saw in the corpus callosum for peak engulfment of OPCs. And indeed, what John showed overall, I won't go through all of the data, but what he showed was that at P0 to P3, again, we have amoeboid microglia accumulating in the lateral ventricle, infiltrating into the corpus callosum, and the fourth ventricle infiltrating into the cerebellar white matter. And during normal expression of CX3CR1, we have engulfment and we have normal myelination, which we think is important for normal connectivity. And when we delete the fractalkine receptor, whether it's in the corpus callosum or in the fourth ventricle, we show the same effect, that there's about a 50% reduction in microglia engulfment of oligodendrocyte progenitor cells, and there's thinner myelin. So we now have 2 areas of the brain being refined at the exact same time, showing that the fractalkine receptor mutant causes an overproduction of oligodendrocytes and thinner myelin. And so I kind of think of it this way with brain development. It isn't the difference between a blob of clay and a refined thinker, Rodin's thinker here. It's actually the difference between refinement here and here. So, there are lots of attributes that are occurring, but it's a difference between refinement of a clearly artistic structure like the thinker versus more of a less artistic, more minimalistic structure here. And we think this is important because diseases like perinatal encephalopathy, which are caused by maternal intrauterine infection or hypoxic ischemic injury, which we know does affect the microglia state, might be altered in diseases where we know that myelination fails despite an increase in the progenitor pool. Also, it's now being recognized that neurodevelopmental disabilities, including schizophrenia and autism, have variants in the fractalkine receptor. And while the majority of those models have been studied as altered synaptic pruning, we think there might be a role based on our data with the fractalkine receptor knockout mutant, that might be a model also for the myelin perturbations that have been documented. So this is something now that Michael Freyshek, a grad student in the lab, is taking on this project to take these developmental studies and explore these roles and how they're being dysregulated in developmental disabilities. And I think what this potentially could present to the field is that there is an unexplored role for pathological alterations in microglia pruning of OPCs, contributing to myelin perturbations in developmental disorders. And we do have an electrophysiologist now in the lab, Rajaram Krishetty, who will be looking at how these myelin alterations are actually impacting neuronal function. And as we all know, proper myelination is needed for action potential propagation, which is necessary for circuit formation. So we want to take this now to understanding neuronal dysfunction. But moving on, we're also very interested in if these early developmental microglia represent a regenerative phenotype of microglia. As we all know, in diseases like multiple sclerosis, we have degenerating myelin. And there has been a lot of studies showing how if microglia don't remove myelin correctly or don't remove debris, that you don't get proper remyelination. So we think that the fact that we have proliferating OPCs, that there might be an unexplored role for the fact that as in development, you always produce too much of a cell. And if that isn't properly refined, perhaps that might be leading to the thinner myelin, which we do see in remyelination. It's never as efficient as it is during development. So we're going to be exploring how we can switch our neurodegenerative phenotype of microglia into a more regenerative phenotype. And if we find a way to prune this oligodendrocyte progenitor pool, would this help with the remyelination in adult demyelinating diseases like multiple sclerosis? Then we moved on in this project to understanding which OPCs are being engulfed by microglia. And in order to do this, we looked at our immunofluorescence staining data that was published originally, and we also explored this at the single cell level. So first off, what we knew from Ashley's paper, again, we took our reporter mice, which are green microglia, crossed with our, in this case, NG2-DS red, so we don't have to induce tamoxifen here. Red is expressed, the fluorescent red color is expressed on these NG2 cells. And then we did a co-localization of immunofluorescence staining with NG2, PDGF receptor alpha, as well as NG2 and O1. And O1 is a marker of developing oligodendrocytes that have not yet myelinated yet. And what we found across these different studies is that overall, NG2 expression is about 88% of the cells being engulfed by microglia express NG2. And we can see that here. So the majority of cells are expressing NG2. Some are expressing PDGF receptor alpha, but that does get downregulated as these cells develop. And then some of those 88% are also starting to express O1, and some of them are starting to express CC1 but less, and none are expressing MBP. So it seems like there's a continuum of proliferating OPCs, and then as they're starting to move into the next stage of pre-myelinating oligodendrocyte, that is when they're being engulfed. So the first question we had with our single cell transcriptomics, and what we do here is we make a single cell suspension of just the corpus callosum, so we can enrich for the amoeboid microglia as well as the OPCs and all the glial cells. And we can look at the transcriptomics of the entire cell. So there's a lot more transcripts in the cytoplasm than there in the nucleus. So in doing this, we not only enrich for the glial cells, but we can see a more sort of broad view of the transcript versus single nuc-seq, which is just going to be in the nucleus, and there's less transcription going on in the nucleus. So if we take a look at the corpus callosum from, this is from pooled five mice at P7, we have a difference of cells here. We do have some neuron progenitor cells, as well as OPCs, and some newly formed OLs, as well as astrocytes, which are categorized here. And what we find is that the majority of cells that express fractalkine are indeed the OPCs that are here. So if we see this yellow OPCs here, see this yellow, and it does get there is less in the OL, so it seems to be less expressed, but the majority is here. And we do have some expression in these excitatory neurons, which would probably be some of the subplate neurons that are left over during early postnatal development that get left out in the corpus callosum. So then we looked at subpopulations of these OPCs, and what we found, which is similar to what Castello Branco has described, is that we have a homeostatic population of OPCs, we have a proliferative population of OPCs, as well as activated OPCs, and then we have pre-OPCs and two populations of microglia. One is a phagocytic population of microglia, and one is a population of microglia that's more homeostatic. And what we wanted to understand is first, looking at a heat map to confirm these populations, which we did here, but also which of these groups is actually expressing the ligand. So it seems, interestingly, that this proliferative population of OPC is most highly expressing the ligand, fractalkine ligand, and to a lesser extent, this homeostatic OPC population is expressing the ligand. Also, we were interested in the don't eat me signal, and that is most highly expressed in OPCs 5 and 4, which are the pre-OPCs. So these pre-OPCs, which then develop into the proliferative, and which we globally call committed OPCs, are expressing this don't eat me signal. And then the receptor for the fractalkine, which is the fractalkine receptor, as well as SERP alpha, which is the receptor for CD47, are both more highly expressed on this phagocytic microglia 1 population than this more homeostatic population microglia 2. So then if we do a trajectory analysis to see how these cells all come together, what we see is that we start off with the pre-OPCs, and that population moves into the proliferative OPCs, and the proliferative OPCs either move into homeostatic OPCs 1 or activated OPCs before becoming the newly formed oligodendrocytes. And what's interesting is that I didn't put it on this slide, is that it seems that the fractalkine, when we pulled these populations out, homeostatic 2 versus homeostatic 1, this homeostatic 1 is actively moving into newly formed OLs. This is the population that's most highly expressing the fractalkine between this population and this population. So this is the population that's getting refined. This homeostatic population downregulates a lot of the committed oligodendrocyte genes. Perhaps they're the satellite OPCs that remain in the white matter undifferentiated until there's a time like an injury when they need to proliferate. So there's a clear sort of segregation of which populations of OPCs are expressing the fractalkine ligand, adding more insight to our immunofluorescing staining during development, showing that there's really two early stages of oligodendrocyte progenitor cells that are being refined. And this is clearly happening before these newly formed OLs occur. And we're not seeing any of that happening in this population of OPCs. So we find that we have fractalkine signaling that's regulating synaptic pruning, which we've shown now regulates the engulfment of OPCs. But there's also been shown that activity-dependent mechanisms also regulate synaptic pruning, but also regulate myelination. And we thought perhaps microglia engulfment of OPCs could be an indirect way that activity is regulating myelination. So is activity of the axon directly signaling to the committed OPC to myelinate, or is there an indirect process that maybe this microglia refinement of OPCs might be playing a role? And so again, what we did is looked now in the third ventricle. So as I mentioned before, the lateral ventricle is where we have this accumulation of amoeboid microglia that eventually infiltrate into this white matter area. And also we see the microglia accumulating in the fourth ventricle that enter into this cerebellar white matter. Well, the last aspect of our ventricular system is the third ventricle. So again, here's our lateral ventricle. Our fourth ventricle is out here where our cerebellum is, and then we have our third ventricle. The third ventricle leads into the optic chiasm, which feeds into the optic nerve. And so again, if we compare the microglia in the cortex versus in the third ventricle, we can see the same morphology of amoeboid microglia emerging at the same time that's occurring in the lateral and fourth ventricle. And so what Irfan did here is did 3D reconstructions across the entire optic nerve to look at how microglia are engulfing OPCs and contacting OPCs. And for the most part, we can see these processes occurring across the entire optic nerve. So each Z-stack is 25 micrometers, so the two put together would be 50 micrometer. So that would be 9 across the entire optic nerve contiguously, which is shown here quantified as OPCs per millimeter cubed, either contacted or engulfed. And so if we do a 3D reconstruction, we can see many different areas again where we have a microglia with an NG2 being encapsulated. And then here, this is the co-localization of these channels, and then added on with bisbenzamide, showing that this cell is indeed a cell. It has a nucleus, a bisbenzamide positive nucleus, as well as the microglia. So a question here is, does neuronal activity affect the engulfment, the microglia engulfment of OPCs? Sorry about that. So what we used is a model that's been used in the past for studying activity-dependent mechanisms, whether it's refinement in the thalamus, synaptic refinement in the thalamus, as well as the cortical columns in the visual cortex. And we asked the question, how do changes in neuronal activity affect microglia engulfment of OPCs during a critical period for synaptic plasticity before eye opening? So really there's 3 critical periods. There's before eye opening. Eye opening happens around P13. Mainly the neuronal activity is through cholinergic processing in the retina. And then you have after eye opening, and then you have later stages of development. So after eye opening, activity-dependent mechanisms are being regulated by light. So before eye opening, there's cholinergic activities of the retina. So what we did is used a model that has been established in the field with TTX. So this is intraocular injections of TTX, which is a sodium channel blocker that blocks neuronal action potentials. And intraocular injections were performed at P5 and P6. So we had intraocular injections at P5, intraocular injections at P6, and then optic nerves were assessed at P7. And what we found overall is that blocking neuronal activity reduced or increased microglia engulfment of OPCs. So as we can see here, these are our control engulfment normalized to 1. And then our TTX treated increased engulfment. So unlike fractalkine receptor knockout, which decreased microglia engulfment, blocking activity caused an increase in engulfment. And this may have some consistency with showing that decreasing activity decreases myelination and increasing activity increases myelination. So if we have less need for myelin, those cells perhaps aren't necessary and they're eaten by the microglia. So when we looked at the number of microglia across this span, as well as the volume of microglia and the sphericity, we didn't see in changes with activity. However, we did view changes in engulfment. And so We also first wanted to look at whether immunofluorescence staining changed fractalkine signaling. So could this increase in engulfment be due to the fact that microglia are having more signal to the fractalkine receptor to engulf those cells? And as a first pass, first experiment, we did show that by immunofluorescence staining in the P7 optic nerve, that the fractalkine ligand is increased. We're not sure where the ligand is actually coming from, what cells are releasing it, and if the OPCs are dead or alive before being engulfed in this activity-dependent paradigm. But this is our first set of experiments to look at how activity of neurons may indirectly regulate this engulfment process that actually impacts myelination. So overall, what I think our data summarizes today is that microglia phagocytose OPCs in the optic nerve during early development postnatally. This is a critical period before eye opening that's been studied for synaptic plasticity. Blocking activity of retinal ganglion cells via TTX significantly increased the engulfment of OPCs, and this suggests that during early postnatal development, fractalkine signaling may play a role in regulating refinement of the OPC pool to indirectly impact myelination. Lastly, I'd like to thank the laboratory. Without all of their hard work, none of this data would be possible for me to present. I'm really excited about the trainees in the lab. They're all doing extremely well, and all are contributing to different aspects of the work. I also like to mention our collaborators at the Mellon Center, but also our collaborators within our department, Antoine Laveau, Nikhil Panikkar, Jessica Williams, and Ho Dana, as well as our funding sources. Thank you, and I'm happy to take any questions.
Thank you, Tara. Fantastic talk. So the questions are going to be handled by Sarah. Sarah, are you there?
Yes, I'm here.
Hi. So you guys can start writing in the chat or raising your hands. In the meantime, if Rosa, do you have a question?
You can start if you
Sarah.
want, otherwise
No, go ahead. Sarah coordinates. Go ahead.
Okay, so Rosa, start.
Okay, thank you. Thank you so much, Tara. Beautiful talk. So I have a couple of questions. Maybe I can start with one about the myelin phenotype, which is very interesting, this myelin perturbation. And I was wondering if you have any sense of how dynamic these perturbations are, if eventually the CXCR1 knockout recover, or if getting worse and worse with time. Do you have any feeling about that?
Yes, I don't, because we haven't done that experiment. I think it would be great to do some later time points, but I think that is a really important question because we have seen in the literature where especially early adolescent, or in this case, late adolescent P30, you can see changes in myelination, but then somehow it recovers. And then does that recovery still leave an imprint on neuronal function or not? So I think probably with the future experiments we have looking, using EFIS to see, are we actually seeing any changes in neuronal function? I think we're going to have to look at some later time points by electron microscopy.
Yeah, thanks. And if I just can connect on this myelin phenotype, if you have, what is your explanation for this reduced myelin? So what would be the mechanism that leads to this reduced myelin?
So we're exploring two things. Actually, we have some preliminary data that I didn't present, but it seems that there are more internodes in these mice than in the wild type mice. But it's actually hard to show that the internodes are increased on the same axon, right? Because it's hard to be able to follow that. I think we're going to have to come up with some more tracing, but we did do the Casper staining with NAV and actually calculated the numbers of internodes that we could count per 3D reconstruction. So it's in the same area. So what we're thinking is that the oligodendrocytes themselves, because they're increased in number, they're still branching and touching the axon. So that might result in an increased number of internodes. So that means the internode distance is going to be shortened. And I think there's been several people now showing that the shorter the internode distance, the thinner the myelin. And some people even saying that the reason why remyelination, remyelinated axons have thinner myelin is because during remyelination, you actually have shorter internode distances. So that's one, I think, sort of structural aspect. I think the other thing we really want to explore is that we know cholesterol, like there's limited resources for cholesterol and other important proteins for the myelin structure itself. And perhaps those resources get spread out so much that you have limited resources to really proper myelinate. So those are kind of the two directions we're going in. But if you have any thoughts, we'd love to hear it, because this is something that we're struggling with to really try to understand.
No, thank you so much. Very, very interesting possibilities. Thanks.
So there is a question in the chat from Guy Brown. So great work. Do you know whether microglia are phagocytizing the intact myelin in addition to the OPCs?
Yes, there's been some interesting studies now. Bruce Apple has shown this in zebrafish. We have not looked at this yet in our model, but we indeed think that probably microglia are also trimming or pruning the myelin sheath itself. I think some people are referring it to as like, I think one paper referred to it as trogocytosis. So you're not actually eating the entire myelin sheath, obviously, but you're trimming or pruning parts of it to make it more efficient and better. So there definitely could be an aspect of pruning of the myelin that's occurring here as well that we haven't explored.
Amanda, yes.
Hi, fantastic talk, Tara. Thank you. I think I found it particularly fascinating, this real clustering of fractal kind within the OPCs. I think that's amazing that they are the major producers of the fractal kind. But I'm not sure if that's sufficient to explain why only a subpopulation of those are culled or pruned, right? There has to be something. Maybe they are dysfunctional, or maybe they because you don't see within that population some of those that may be signaling that they have to be killed by microglia. So how do you understand this? How do microglia know which ones? Why wouldn't they eat all of them?
Right. Yes. So our best way of looking at this so far is we're digging deeper into our single cell transcriptomics to try to look to see for some pathways. And you're exactly right. We totally agree, because we see microglia contacting an OPC, and it's almost like when it contacts it, it's making a decision, should I or shouldn't I eat it? And so is the OPC itself like it's looking for that signal, and if it doesn't get it, it's moving on to something else. So we definitely don't think it's all fractal kind. We think that there's something else involved. But I guess what's unclear is like, let's say it's another upregulated signal in addition to the fractal kind. Is that coming from the activity of the axon? So that OPC happens to be closer to the axon, so it's going to get that signal and it's going to upregulate it. And then that's going to tell the microglia, not to eat it. So I think what we think about with the fractalkine receptor is it's classically not considered a phagocytic receptor, but perhaps it's needed to get the cells in close enough contact so they can activate phagocytosis or just the sensing and moving on. But yeah, we definitely think there's something else going on there.
Yeah, I think that this point of the activity of the neurons is interesting. Do you conceive any possible like in vivo imaging experiments where you can visualize which OPCs are more active or less active, and see if that correlates with the probability of being engulfed by microglia or not? Is that feasible, or is it just something like, does it make any sense to you?
Yes, so we started doing some ex vivo imaging of the optic nerves. So you can actually, like, you can make a coronal slice for the brain, like we did the ex vivo imaging to look at the mitochondrial membrane potential. We've started to do this where you take the optic nerve out and ex vivo image it and look at, we can look at the optic nerve alive. So what's nice about the P7 optic nerve is because it's not myelinated, you can do that. The only issue, it's a little bit of a technical issue because with the section, we can use harps to keep it in place for the ex vivo imaging. For the optic nerve, it's a little bit harder and sometimes the optic nerve moves. So we still have to work on that. And so we thought we would focus on the perfusion fixed tissue and the EM first, since that's, the easier way to go. The other way we're thinking about it too is using flow cytometry and using different markers to look at different populations. And then the OPCs that aren't engulfed, we can FACS sort those and maybe do some proteomics on them and see what differences they may have.
Okay, thank you. Great. Looking forward to those experiments.
Oh, thank you.
So there's two other questions. The first one, is there any involvement of the complements?
I'm sorry, can you repeat that?
Is there any involvement of complements?
Yes, we have not looked at complement yet, but that's another receptor that obviously was shown to be involved in activity-dependent synaptic pruning, but we don't have any data on that yet.
Okay, and the other one is, any idea of the metabolic status of the phagocyted OPCs?
So the OPCs that are being engulfed, what's the metabolic state?
Yeah.
Oh, we have not looked at that, but now that we've got our single cell data, I guess we can start to explore that, explore those states.
Yeah, Rosa.
If I can ask another question. So regarding to the activity data with the TTX experiment, which is super nice to see that fractalkine itself is upregulated. Have you tried this in the CXCCY1 knockout? Do you abolish?
No, but yes, that is what we want to do. So if we do the same experiment in the knockout mice, do we block that increased engulfment? And that would actually really tie in the mechanism. But yes, that is a plan we have.
Yeah, it's cool. Yeah.
Just one more.
Yeah.
Do you think this developmental regulation of OPCs, could it be similar to remyelination during disease, or do you think it's completely different? Or
Yeah, we're looking into that because perhaps the microglia, what we think, are distracted in clearing myelin debris. And there's also lots of inflammatory processes going on. And
right.
so we are hypothesizing that the microglia, the OPCs, are not being refined at all during disease states. But that's something that Isabel is taking on as her thesis project. And so hopefully she'll provide us insight into that.
Great, thank you. Any other questions from the team members or the audience?
There is one from Izaskun, if I'm seeing right. Yeah.
Thank you very much for your incredible presentation. A little bit related to Amanda's question. Have you seen in different OPCs population in an EA model or another multiple sclerosis model the same alteration in fractalkine expression or not?
Oh, are you asking, I'm sorry, are you asking, does fractalkine expression increased in any of the EAE models or in the any models of MS?
Increased or at least different expressed in different subpopulation of OPCs?
Yes, we have not looked at that yet. So Isabella is starting to look at that in one of the animal models of remyelination for MS. So she'll be able to give us some insight on that, but I don't have any information on that yet, whether fractalkine is differentially expressed in those OPCs or might be regulating. What's been published is that the fractalkine receptor is regulating microglia engulfment of myelin debris. But whether it's regulating any type of OPC engulfment, we don't know yet.
that's why if I have a 10 seconds for a last question. It
Yeah,
was
go ahead.
related to what you have just said. The experiments that have been done with Plexicon, for instance, or other microglial depletion experiments in EA model, do you think that it's because of myelin engulfment or because of or OPC effect?
Well, I think microglia are doing a myriad of things, and that's why we wanted to use selectively deleting the phagocytic receptors to look at changes, because Margot Prince and others have shown that microglia release factors that is necessary for myelin development. So I think when you completely remove myelin, you may get a different phenotype than selectively inhibiting the ability of microglia to engulf or phagocytose. So there could be multiple things. Plus, microglia themselves contribute to the inflammatory environment as well. So removing them, you may indirectly dampen inflammation, which could also have positive effects on myelination and development.
Thank you.
Yep. I think we don't have any more questions. Sarah?
No.
Okay, great. So it was an amazing talk, great discussion. Thank you very much, Tara. And thanks everybody for showing up. Just remember, next talk, we will send you a different link, but this will be explained on the email. So thank you everybody and see you next month. Bye-bye.
Thank you so much for the feedback. It was a really great discussion. I really enjoyed talking with everyone. I hope our paths cross again, or maybe down the road as we get more data, we can come back and share with you guys. I'd love to get your insights.
Sure, absolutely.
Thank you.
Absolutely.
Bye.
Thank you very much, everybody.
you.
Bye-bye.
Bye.